Welcome to Best of Biology. Today the topic that we are studying is heredity. Do you know why is it when a baby is born, people start comparing if the baby looks similar to their father or to the mother. Also, why is it that the babies look similar to their parents? This is because characters are transferred from parents to their children, and this passing of characters from parents to children is called as heredity. Heredity is also known as inheritance. The characters that are passed from parents to children must be in the form of some information. This information is carried in gene. Thus, gene is what? Gene is the unit of heredity that carries the information that is inherited in an individual. What does gene consist of? Gene consists of DNA. DNA is a chemical compound, deoxyribonucleic acid, and DNA consists of three things. One, a ribose sugar. Secondly, a phosphate molecule, and third, nitrogenous base. Nitrogenous base is nitrogen containing biological molecule. Genes are present in the nucleus of our cells. There are around 20,000 genes present in every cell of our body. Think, why is it that some children look much identical to their parents and others do not resemble at all? Why is it that just a specific characteristic is visible in a child while other does not carry it? For example, one child in the family carries the genetic disorder like diabetes while other does not carry it. To understand all this, a scientist called as Gregor Johann Mendel, also popularly known as father of genetics, conducted a variety of experiments in a plant called as garden pea, or scientific name of it is known as Physon sativum. Mendel's work was widely accepted because he proved his results by combining statistics and mathematics with science. To understand Mendel's experiments, we need to understand some important scientific terms that are related to heredity. First is genetics. The study of biological inheritance, that is the study of passing of characters from parents to offspring, is called as genetics. Progeny is another term for offspring or children. Third is trait. Trait means a specific characteristic in an individual. For example, if we talk about characteristic height, so the trait of this character is either tallness or shortness. Fourth is alleles. Different forms of the same gene are called as alleles. Mendel, by his experiments, told that both father and mother contribute equal amounts of DNA in the child. That means every trait in the child is influenced equally by both father and the mother. If we consider the trait height here, the genes can be represented by letters, and we are considering father to be tall heighted, so genes can be represented by capital H, and mother to be short heighted, whose gene can be represented by small h. So the child will receive one copy of gene from both the parents, that is here one capital H from the father and one small h from the mother. So these copies of gene are called as alleles. Alleles are represented by alphabets. Fifth is genotype. Genotype means the genetic makeup of an individual. It is the collection of genes for various traits. Genotype is represented by a set of alleles for a particular trait. That means in the above case, this capital H and small h, that is the set of alleles, is the genotype of this child for the trait of height. Next is phenotype. Phenotype means the physical characteristics in an individual. For example, that is the external appearance. For example, if the child appears to be tall or short-heighted, if he has black eyes or brown eyes, so that is the phenotype of the child. Next is dominant allele. The allele that is phenotypically expressed. What does this mean? In the above case, we saw that the child receives two copies of gene. So, in this child, if the gene for the allele for the height, tallness is visible in this child, that is the child appears to be tall, then we can say the allele for the trait tallness is phenotypically expressed, that is visible in the child, and that particular allele will be called as dominant allele since it is phenotypically expressed. Dominant allele is represented by the capital letter. Recessive allele is the another allele whose effect is not seen in the child. That means it is hidden or masked. Recessive alleles are represented by a small letter. And please keep in mind that the recessive allele is just hidden character. So it can express itself if the dominant allele is absent. Next is homozygous. An organism is called to be homozygous if it receives copies of similar alleles. That is, this father has similar alleles for the trait height, that is capital H. So he has received similar copies from his parents. So he has received capital H and capital H from both the parents. And he is, since these are similar alleles, so it is homozygous. Homo means same and zygous means zygote. So copies of similar alleles are called as homozygous. Heterozygous means if the organism receives different alleles from both the parents. Like in the case of this child, he has received different alleles, one capital H and one small h. So this child is heterozygous. Now let us understand Mendel's studies. Firstly, some students might think that why Mendel did not work on humans. So think experimentally, is it possible to study inheritance pattern in humans? I mean, humans take years to grow and form offspring. So they have a short lifespan and it is not possible for anyone to study many generations. Maximum 3 to 4 generations only can be studied. So this is not possible to conduct an inheritance study on humans. Secondly, there cannot be a controlled study on humans. I mean, if the number of offspring has to be studied and if a particular trait has to be studied. So quality of traits cannot be controlled in humans. Secondly, why Mendel chose the plant? There are many reasons for it. Firstly, it has short lifespan. So it is possible to study many generations in this plant. Secondly, it is easy to grow. And thirdly, there are 7 pairs of contrasting characters. Actually, there were many contrasting characters present in the pea plant, and of which 7 pairs of different traits, that is contrasting characters, can be easily manipulated by Mendel. So he chose these 7 characters for the pea plant. So let us understand what were the traits that Mendel studied. First is flower color. So flower color was either violet or white colored. Second, flower position. Flower position was either axial, that means around the axis, along the stem, along the length of the stem, and either terminal, that means flower was at the top. Seed shape. Seed shape could be either round shape or wrinkled, that there are wrinkles in the seed. Fourth is seed color. Seed color was either yellow or green. Pod shape. Pod means in Hindi we call it fali of the pea plant, matar ki fali. So seed color, pod shape was either inflated. Inflated means it is swollen, fuli hui fali, or either constricted, that it is contracted. Pod color was either green or yellow. And stem length, the length of the stem was either tall or short. So these were the 7 traits that Mendel studied. Mendel studied he crossed and mated around 29,000 pea plants in his complete study, and he counted exact number of progeny plants. And what he observed, that the 7 traits produced almost similar pattern. What was that? The similar pattern was that one trait always concealed the other trait in the first generation, and the trait that was expressed was called as the dominant trait. So here he called these traits as dominant because these all traits were expressed in the first generation of the progeny, and these all were called the recessive traits since these were hidden in the first generation. Let us understand now Meadows experiments. I told you that Mendel crossed around 29,000 plants, and for a particular trait, for a single trait, he crossed around 1000 plants. Firstly, he crossed plants for a single trait. That is, he chose a trait, stem length, that is length of the stem, to be tall heighted or short heighted. Short heighted is also known as dwarf, that is tall plants crossed with dwarf plants. The cross for the single trait was called as monohybridcross. That means mono means single, hybrid means mixing. So mixing for a single trait was called as monohybridcross. So when Mendel crossed tall plants with dwarf plants, what he observed was that in the first generation, all plants came out to be tall. That is, the tall plants were dominant over dwarf plants. P means the parent generation, and first generation from parents was called as first filial generation, that is F1 generation. Next, he allowed the F1 generation plants to be self-fertilized. That means here tall plants are represented by capital T, and dwarf plants are represented by small t and small t. So all plants came out to be tall with genotype capital T and small t. So he allowed these plants to self-fertilize, also called as selfing. Self-fertilize. When they were self-fertilized, what result was observed? They were crossed by a method known as Punnet's square. Punnet's square was found by scientist Punnet and it was named after him. So this is the method of the Punnet's square. How it is done? One genotype is placed horizontally, another genotype is placed vertically. How to observe now? This capital T, small t crossed with capital T and small t. See the results. Capital T, capital T, capital T, small t. Capital T, small t and small t from here, one small t from here. So this was called as F2 generation, and the results that were observed was in the ratio 3 tall and 1 dwarf plant. Capital T were considered as the tall and the dwarf was considered for the small letters. So this was the phenotypic ratio. That means it was the ratio that was observed in the plants and we call it phenotype. Okay. So next, how is the genotype ratio calculated? We will write them. We will write all the possibilities of the genotype. One, capital T, small t. Second possibility is capital T and small t. And next is small t and small t. Count them. Capital T are 1. Capital T and small t are 2. Here 2 and small t are again 1. So this is the genotypic ratio of the monohybrid cross. That is 1:2:1. The original numbers of the Mendel's studies were 1000 plants for the single trait, and out of which 760 were observed to be tall and 230 around were observed to be dwarf. So the ratio that was calculated for the phenotype was 3:1 and for the genotype it was 1:2:1. Let us see now what results came when Mendel selected 2 traits for his experiments. That is a dihybrid cross. Di means 2 and hybrid means mixing. So mixing of plants for 2 traits. The traits that Mendel selected were seed shape and seed color. The shape of the seed was either round or wrinkled and the color of the seed was either green or yellow. Notice that the color of the seed yellow is dominant over green seed color and round seed is dominant over wrinkled seed. Dominants are represented by capital letters and recessive are represented by small letters. So these were the results of the F1 generation. These are the parent generation and these are the F1 generation in the Mendel's experiments. After F1 generation, Mendel self-pollinated the F1 plants. You can notice that the F1 generation received one copy of allele from both parents. One capital R from here and one small y from here. One small r from here and capital Y from here. So after F1 generation was obtained, they were self-pollinated. This cross can be done with the help of Punnet's square that we have discussed earlier. You can draw this Punnet's square in your notebook so that to understand this cross becomes easier for you. You can do it by yourself. Just see here. These are the copy of the alleles that were received by F2 generation. That is, the seed can be either round yellow, wrinkled yellow, round and green, wrinkled and green. So when these alleles were self-pollinated, what were the results? Let us do with the help of Punnet's square. Capital R from here and capital R from here. Capital Y from here and capital Y from here. Here, capital R from here and small r from here. Capital Y from here, capital Y from here. Similarly, capital R, small y, capital Y. Capital R, small y and capital Y, small r. Capital R, small r, capital Y, capital Y. Small r, capital Y, capital Y. Capital R, small r, capital Y from here and small y. Small r, capital Y, small y. Capital R and capital R, capital Y and small y. Capital R, capital Y, small y. Similarly, you can find out for all of these possibilities. Let us see now what are the results in the phenotypic ratio and genotypic ratio. Let us see how the results were obtained for a dihybrid cross. That is the results of the phenotypic ratio and genotypic ratio. For phenotypic ratio, there are 4 possibilities that we discussed. One is round yellow denoted by capital R, capital Y. Second is wrinkled yellow denoted by small r, capital Y. Third possibility is round green. So round represented by capital R, green by small y. And fourth possibility is wrinkled green. So both are recessive characters and are represented by small r, small y. So when we will count them from here, what are the ratios that are obtained? Round yellow occurs in the ratio of 9, wrinkled yellow R3, round yellow R3, round green R3 and wrinkled green R1. So 9:3 is to 1 is the phenotypic ratio that is obtained in the dihybrid cross. And how is the genotypic ratio obtained? It is obtained from here, from the set of alleles. For example, 1 here is for capital R, capital Y, capital Y. 2 is for this one, capital R, small r, capital Y, capital Y. So you have to count these. Same alleles should be obtained. So here, one is here and second is here. So these are the 2 genotypes similar. So we are giving them a ratio of 2. Similarly, you can find out for all the alleles, and the ratio, genotypic ratio that is obtained for dihybrid cross is 1:2:1, 2:4:2, 1:2:1. So this complete is the genotypic ratio for the dihybrid cross. Scientist Mendel, on the basis of his experiments and the results obtained from the monohybrid and dihybrid cross, he came out with the rules of inheritance, and we will discuss them in our next video, that how we analyzed from these results the rules of inheritance. Also, we will be discussing test cross, back cross and sex determination in humans. Thanks for watching.